Hello everyone, and welcome to Biopedia. This week, I thought I'd do another rewind episode and focus on a topic we've mentioned a few times now, but I haven't really covered in great detail: proteins. Proteins are vital for life as we know it, and are ubiquitous throughout the tree of life. However, in order to properly understand proteins, we need to look at the subunits that they consist of, amino acids. In many ways, a protein can be thought of as a string of beads. Each amino acid would be one bead on the string. The sequence of amino acids is also important for determining the protein that will eventually result. As we'll discuss below, each amino acid has a slightly different functional group, which can impact how it interacts with other amino acids and thus help determine the structure. An amino acid is an amphoteric molecule, meaning one end of it is acidic and one end is basic. Specifically, it has both a carboxylic acid group, with the chemical formula COOH, and an amine group, with a chemical formula NH2. Both of these functional groups are attached to a central carbon, which also contains one hydrogen and one variable group, which is what makes different amino acids distinct from each other. For example, the amino acid glycine has a hydrogen in this available spot. When two amino acids bind, these different groups, the COOH and the NH2, react, and a water molecule is ejected, leaving a bond that goes directly between the carbon on one amino acid and the nitrogen on the other, a so-called peptide bond. It is the interactions between these variable side chains, the groups mentioned above, that are crucial in determining the shape of a protein. Different functional groups have different properties. For example, some of them are acidic and some are basic. If you're interested, there'll be a link in the description to a web page containing the functional groups of all 20 common amino acids. Although we won't go into detail in this now, side chains with different properties accordingly interact with each other in different ways. Different interactions ultimately determine the shape of the protein by either attracting, repelling, or bonding with each other to create a specific conformation which is based on the sequence of amino acid and thus the order of these interactions. In this way, the sequence of amino acids in a protein, the so-called primary structure of the protein, can determine the shapes that subsequently form, such as helices or pleated sheets of amino acids. This is the secondary structure. The interactions between side chains and secondary structure domains ensure that the protein folds in a particular way, creating its specific tertiary structure. There is a final layer of complexity above this, the quaternary structure, or how different protein subunits interact with each other to form a larger structure. A good example of this would be hemoglobin, which consists of 4 subunits, each of which combine to oxygen. However, all 3 layers of complexity above the primary structure rely on the interactions between amino acids that are determined by which of the variable side chains, or R groups, to put another way, interact, and where they are in the protein sequence. In nature, there are 20 common amino acids. Most animals can synthesize about half of these if their diets contain sulfur and nitrogen. The rest, which can only be obtained ready-made from their diets, are so-called essential amino acids. Other amino acids are possible other than these 20, but these are never encoded for in naturally occurring biological systems. We will have to leave the discussion of how amino acids are coded for, however, along with the specific interactions between R groups, for another time. In the meantime, if you have any comments, questions, or topic suggestions for future episodes, feel free to get in touch at biopediapodcast@gmail.com. Thank you for listening, and until next time, have a great week.